The bald eagles were found on this farm in Federalsburg. State officials have a working theory that the eagles may have been poisoned, but the person who owns this property says there is no reason for poison to be there.
Something was killing America's bald eagles, and at first, no one could explain it. In 1994, officers at DeGray Lake found eagles dead along the shore, others still alive, shaking in the water, unable to stand or fly. No bullets, no broken wings, no signs of attack. Then scientists looked inside their brains and found tiny holes. For 27 years, every theory failed, until investigators traced it to a hidden toxin in the food chain, one that survived cooking, reached fish people were eating, and forced officials to close the lakes. Dead eagles at DeGray Lake. In winter 1994, wildlife officers at DeGray Lake in Arkansas found bald eagles lying along the shoreline. Some were already dead. Others were still alive, but their bodies were failing right in front of the officers. Their heads drooped, their legs buckled. They trembled in the shallow water and stumbled in circles, unable to lift off from DeGray Lake, even though their wings looked intact. Officers checked the bodies for the obvious signs. No gunshot wounds, no broken bones, no torn feathers, no predator tracks near the Arkansas shoreline. These bald eagles were not starving or weak. They looked healthy on the outside, which made the scene even harder to understand. Whatever was killing them was not attacking their wings, bones, or muscles. It was attacking the nervous system. Because bald eagles were federally protected after the DDT crisis of the 1950s to 1970s, this could not stay a local wildlife case. Federal officials were brought in within days. Pathologists collected tissue samples and searched for lead poisoning, pesticides, infection, or trauma. But in those first reports from DeGray Lake, there was only one clear pattern. The birds were losing control of their bodies before they died. That strange winter die-off in Arkansas became the start of a mystery that took 27 years to solve. Microscopic holes in the brain. When pathologists studied the bald eagles from DeGray Lake after the 1994 die-off, they did not find wounds on the body, broken bones, or an obvious poison in the stomach. The real damage was inside the brain and spinal cord. Under the microscope, the white matter was filled with tiny empty spaces, called vacuoles, and it was not just one bird. The same tiny holes appeared in the same white matter tissue again and again. Scientists named the condition vacuolar myelinopathy, or VM, but the name only described the damage. It did not explain the source. The disease was damaging myelin, the protective coating around nerve fibers. Without myelin, the brain cannot send clear signals to the body. That is why the eagles stumbled, shook, lost balance, and could not fly properly. Their bodies were alive, but the signals between the brain and body were failing. What disturbed researchers most was how consistent it was. Every tested eagle showed the same damage in the same part of the nervous system. That meant this was not random sickness. Something from the DeGray Lake environment was entering the birds and attacking their nerve tissue. The USGS, EPA, CDC, and university teams had no known disease, poison, or injury that matched what the microscope showed. They had a name for the damage, but they still did not know what was creating these holes inside the brains of bald eagles. One lake becomes 10 states. Within 2 years of the 1994 DeGray Lake die-off, the same strange deaths appeared at Lake Moultrie in South Carolina. Bald eagles were shaking, stumbling, and collapsing just like the birds in Arkansas. When pathologists checked their brain tissue, they saw the same tiny holes. That meant DeGray Lake was not an isolated event. Then the map started filling in. Georgia, North Carolina, Tennessee, and more reservoirs across the southeast and Midwest. But the eagles were only the most visible victims. American coots, small diving birds that eat underwater plants, were dying in much larger numbers. Mallards, Canada geese, and ring-necked ducks were also showing VM damage. That gave investigators their first real clue. Eagles hunt coots. If coots were eating something toxic from the lake, every eagle that ate one could be taking in a much stronger dose. By the early 2000s, vacuolar myelinopathy had been found across at least 10 states. The strangest part was that many reservoirs were not connected. Some were hundreds of miles apart. So this did not look like a disease moving lake to lake. It looked like the same hidden condition was appearing in separate places, quietly moving through the food chain, while the USGS, EPA, and university teams still could not name the killer. 27 years of failure after the 1994 deaths at DeGray Lake, scientists started with the most likely causes of eagle deaths. The USGS, EPA, CDC, and university teams tested pesticides first, because bald eagles had already been devastated by DDT during the 1950s to 1970s. If another chemical was damaging their brains, researchers expected to find a known pesticide in the tissue. But the results came back clean. No pesticide matched the holes found in the eagle's nervous tissue. Then, researchers tested for heavy metals. Mercury, lead, cadmium, and arsenic were checked in bird tissue and lake samples, especially around reservoirs near farms, factories, and older industrial areas. These metals can collect in fish, birds, and sediment over time, so the theory made sense. But the metal levels were too low to explain the shaking, the collapse, or the brain damage seen under the microscope. Next came known freshwater algae toxins, microcystin, toxin A, saxitoxin, and cylindrospomopsin. These poisons can kill wildlife in lakes, so researchers tested for them during active die-offs. Still, nothing matched. Year after year, eagles died, and lab results ruled out one cause after another. By the 2000s, the investigation had lasted nearly 27 years, cost millions of dollars, and eliminated more than 40 theories. The real toxin was not appearing in any standard test used during the investigation. Dr. Susan Wild's 16-year pursuit. By the early 2000s, the VM investigation had already passed through several research teams without identifying the cause. Grant cycles ended, federal teams changed, and new wildlife emergencies pulled attention away. But at the University of Georgia, Dr. Susan Wild stayed with the case. She was a wildlife ecologist at the Warnell School of Forestry and Natural Resources, and she studied how freshwater plants, birds, bacteria, and lake sediment interact inside one food web. For the next 16 years, Wild kept following the trail while tests for pesticides, heavy metals, and known algae toxins kept coming back negative. Instead of repeating the same failed panels, she changed the investigation from testing known poisons to tracing the food chain backward. If American coots were eating underwater vegetation and bald eagles were eating infected coots, then the real question was not only what killed the eagle, it was what the coot had eaten before the eagle ever touched it. That led Wild to the plant surfaces inside affected reservoirs, especially hydrilla and other submerged vegetation where birds were feeding. The evidence was not dramatic. It was scraped from wet leaves, examined under microscopes, and tested through slow lab work. Wild focused on the first step in the food chain, the surface of the plants eaten by coots. For years, researchers had treated those leaves as background. Wild treated them as the crime scene. How an aquarium species broke the food chain. The plant Wild kept finding was Hydrilla verticillata. At first, it looked like ordinary aquarium greenery, not the start of a wildlife crisis. Hydrilla is native to Asia and parts of Australia, and it likely entered the United States through the aquarium trade in the 1950s and 1960s. When people dumped unwanted tank water into canals, ponds, or lakes, broken hydrilla fragments escaped. One small piece could grow into a new colony. Once hydrilla entered a reservoir, it was hard to stop. The plant spreads by fragments, so boats, currents, and dumped aquarium water could carry it from one place to another. Under good conditions, it can grow more than 1 inch per day, forming thick green mats across the water. Those mats block sunlight, crowd out native plants, reduce oxygen, and give diving birds like American coots a steady food source. Then Wildy's team compared affected reservoirs with unaffected ones. The pattern was clear. Confirmed VM sites had hydrilla. Reservoirs without hydrilla had no confirmed VM cases. After 27 years of failed toxin tests, hydrilla became the first consistent link across the mystery. Coots were feeding heavily on those leaves. Bald eagles were eating the coots. So if something toxic was growing on hydrilla, the path was simple and deadly. Plant to coot, coot to eagle. A fish tank plant had helped open a toxic pathway through American reservoirs. A killer that had no name. When Dr. Susan Wild and her team scraped the surface of hydrilla leaves from affected reservoirs, they finally saw what everyone else had missed. The leaves were not clean. They were coated with a thin layer of cyanobacteria, a tiny photosynthetic organism that can live on plants and sometimes produce powerful chemicals. This same strange coating showed up on hydrilla from lakes with VM deaths. But when the team checked hydrilla from lakes without the disease, the organism was missing. After nearly 27 years, the investigation had finally found something that belonged only to the danger zones. Then came the part that made the discovery even stranger. Wilder's team pulled genetic material from the organism and compared it against international biological databases. It did not match anything. Not a known lake bacteria, not a known toxic algae. Not a species already described in science. This thing had been sitting on underwater plants in American reservoirs for years, possibly decades, while researchers tested everything around it, except the one surface it was living on. It was not hiding deep underground or in some remote jungle. It was sitting on leaves eaten every day by American coots. The team gave it a name, attack.Then hydrilla. The meaning was blunt. Eagle killer that lives on hydrilla. It sounds dramatic, but in this case, the name was almost a case report. Attack.Then pointed to the dead eagles. Hydrilla pointed to the invasive plant. Together, they described the whole path. Bacteria on hydrilla. Hydrilla eaten by coots. Coots eaten by eagles. For the first time, the mystery had a living suspect. The discovery was published in Science in August 2021, one of the most respected journals in the world. That mattered because this was not just another lake toxin. Researchers had found a brand new species producing a brand new poison inside ordinary American reservoirs, and it explained why the case had defeated scientists for so long. Every test from 1994 onward was looking for something already known. But this organism was not in the books. No standard test could find it because no one knew it existed. The killer had no name because science had never met it before. The toxin that survives fire. Once attack.Then hydrilla was identified, researchers still had to answer the most important question. What chemical was this new cyanobacteria producing on hydrilla leaves? The answer was a newly identified neurotoxin called attack.Then toxin. It was not a pesticide, not a heavy metal, and not one of the known algae poisons scientists had tested for since 1994. It was a brominated compound that had not been found in lakes, animals, plants, or lab records before. Controlled lab tests showed the toxin could produce the same VM lesions found in dead eagles from DeGray Lake. The same tiny holes appeared in the same nervous tissue. The same damage showed up in the brain and spinal cord. That confirmed attack.Then toxin was not just present at affected reservoirs. It could cause the disease. After 27 years, scientists finally had the compound behind the neurological collapse. Then came the most alarming test. Researchers heated contaminated fish tissue at temperatures similar to pan frying, grilling, and baking, around 350 degrees Fahrenheit. The toxin survived. It did not break down with ordinary cooking. That created a possible human exposure concern, because attack.Then toxin had been found in the edible muscle of fish from affected reservoirs, the fillets people actually eat. A person could catch fish from an affected lake, cook them normally, and still consume the active toxin. The final clue opened another problem. Attack.Then toxin only forms when bromine is present. Freshwater reservoirs normally do not contain enough bromine to trigger that process. So if the toxin was forming across multiple states, an outside source was likely introducing enough bromine to activate toxin production. That meant the mystery was not fully solved. The killer had been named but the trigger was still out there. The evacuation, advisories, closures, and 27 years of silence. After the science paper was published in August 2021, the findings moved from research papers into health advisories at affected reservoirs. State agencies and the EPA began issuing warnings across parts of the southeast. Some areas were restricted, some recreation was limited, fishing notices started going out, and the warning was no longer only about dead bald eagles. The toxin had been found in fish muscle, the part people cut into fillets, cook, and eat. That shifted the concern from wildlife deaths to possible human exposure through fish meals. For 27 years, people had fished in lakes where VM was already moving through the food chain. They caught bass, cleaned the fillets, fried them, baked them, grilled them, and believed the heat would destroy anything dangerous. But toxin survived cooking. A fish could be fully cooked, browned, and served like any normal lake fish, while still carrying the same compound linked to brain damage in eagles. These were not small private ponds with little public use. DeGray Lake in Arkansas receives large numbers of boaters, campers, and anglers each year. Lake Moultrie in South Carolina covers about 60,000 acres and supports heavy recreational fishing. Between 1994 and 2021, millions of fishing trips happened across affected reservoirs. Families camped there. Anglers filled coolers there. Local towns depended on fishing, boating, camping, and lake tourism. But for most of those years, people had no clear warning about this specific toxin because officials still had not identified it. The remaining public health question is specific and unresolved. What happened to people who ate fish from those waters before the advisories came? So far, no major federal study has clearly shown whether long-term, low-level exposure to toxin harmed humans. Maybe the doses were too low. Maybe human bodies process it differently. Or maybe no one has looked closely enough. The bird mystery finally had a name, but the effects of 27 years of possible low-level exposure in people had not been clearly studied. Why this is not over. The 2021 discovery identified toxin, but it left researchers with one major unresolved problem: the source of bromine. The toxin does not form unless bromine is present. That matters because normal freshwater reservoirs in Arkansas, Georgia, and South Carolina usually do not contain enough bromine to allow Etoxinus hydrilicola to produce the brain-damaging compound. Environmental researchers identified 3 possible bromine sources: industrial chemical discharge, coal ash ponds, and agricultural runoff from brominated pesticides or soil treatments. Any of these could carry bromine into lakes through streams, groundwater, or storm runoff. But no single source has been officially confirmed across all affected reservoirs. That means scientists know the toxin needs bromine, but they still do not know exactly where the bromine is coming from. That turns the case into a question of human responsibility. If bromine came from industrial waste, coal residue, or farm chemicals, then VM was not just a natural wildlife disease. Hydrilla gave the bacteria a surface to live on. American coots ate the hydrilla. Bald eagles ate the coots. But bromine may have provided the chemical condition needed for toxin production. Without it, the bacteria might remain on the plant without making a toxin. The investigation remains unfinished because the bromine source has not been confirmed. Hydrilla is already present in at least 30 states, and the bacteria has been found outside the original danger zone. The plant is spreading. The microbe is spreading. The food chain is already in place. If another reservoir receives enough bromine from coal waste, factory discharge, or farm runoff, the same toxic pathway could start again. The toxin has a name. The bacteria has a name. The plant has a name. But the source of the bromine is still the unresolved part that determines whether more lakes could become toxic.